Either you have a piece of DNA with nothing on it, or you have the same piece of DNA with a receptor on it. Does everybody see these are the two choices that you can see? And then what you do is you run this in an electrophoretic gel. You guys have electrophoresis, right? So in electrophoretic gel, you put it in electrophoresis that separates by size. That's a native, it's a native gel because it maintains, and this was a tricky part of this development about 30 years ago, you maintain the GR DNA or the GR-GRE interaction. Very, very hard, tricky part of this. And this separates by size. Okay? So let's think about this. In this experiment, the only thing you can see is the radiolabeled DNA. And we're going to use these erasers as the GREs. Here's two GREs. You're running in the experiment, right? All you can see is the erasers. That's all you need to think about right now. See the erasers. One of these erasers has the water bottle on it. This is the GR. Which one of these molecules is bigger? That one, right? Whoop, not anymore. Mutated GRE, I guess. Okay? So if you run the electrophoretic gel, if I can do this easier. There you go. So you run the electrophoretic gel, all you can see is the erasers. Same size DNA, right? You start running the gel, and this guy runs further down the gel because it's smaller. Does everybody see that? So if you have a band that goes all the way down, there's no binding. If the band gets held up in the gel, guess what? You got something bound to it because it separates by size. Everybody see that? But again, the only thing you can see is the erasers. You cannot see the water bottle in this case. Okay, this is your radiolabeled GRE. So that's how this works. I'm saying it in a very straightforward way here, but this took probably 5, 6, 7 years to get developed properly. Where? Yes? No, the glucocorticoid receptor has something called a zinc finger, which binds, its role and design is to bind to DNA. Good question. Well, then you're getting at the mechanism of binding. If you guys are interested in this a little more detailed, I teach a hormone action class in the fall that gets into this in a pretty serious way, okay, experimentally, point of view. But it's a very good question. It has something called a zinc finger, and it binds to the major groove of the DNA and recognizes specific. So it comes down to amino acid, residue, DNA base interactions that create the affinity for the binding. Okay, good question. So all you can see is the erasers, right? All you can see is the size. So let's run this gel out. Here's your electrophoretic gel. So here we just run the GRE. And it runs, you see a band on a gel, just like you see the eraser with a band on a gel. This is just the radiolabeled GRE. So then you have the GR plus the GRE. And what would happen to this band? It should not migrate as far, right? Because larger molecules stay up in the gel and smaller ones come here. Everybody see that? So where would this run? The same distance, faster or slower than just the DNA itself? Slower, exactly. So it runs slower. Okay, so now we have the basis of an assay. Now we have the GR to ERE. Oh, excuse me, let me do it the other way around. That works also, but ER to the GRE. Remember, we're going to talk about the estrogen receptor if it's still up here. Yes. The estrogen receptor binds TCA, TCT. Again, you don't need to know that sequence. Would this bind to the GRE if I told you this sequence was exquisitely important? Should it bind or not? Should not. So what would the band look like on the gel? Would it look like here? A, B, or C? A, it would look like it would run like there's no binding. Great, that's exactly right. So here's your ER, here's your estrogen receptor. Doesn't bind properly. So all you get is that straight DNA. Any questions? You can do the same thing with estrogen receptor and the ERE and get the reverse results. In that the estrogen receptor binds the ERE, but not the GRE. Does everybody see that? GR is specifically binds the GRE, ER binds the ERE. Because we're going to complicate this very much so in a second. Okay, so if you had this assay going, what would be one thing you might want to do? You might want to try every receptor you can get your hands on and see what happens, right? So they then looked at the androgen receptor to the GRE. Let me just extend our gel here a little bit. They looked at the mineralocorticoid receptor in the GRE. They looked at the progesterone receptor in the GRE. Okay? And what's your first thoughts? Do you think these other receptors should bind the GRE if there's any sort of sequence-specific interactions? You think maybe not, right? And people were willing to bet the farm on this. Fortunately, most of us in the field did not have a farm, so it didn't matter. Turns out, all of these guys competently bind the GRE. The mineralocorticoid receptor recognizes a TGT, TCT. The androgen receptor, the same thing. The progesterone receptor, the same thing. Big time surprise in the field. But think about the responses. Progesterone is involved in pregnancy, menstrual cycle regulation, estrogen, testosterone rather is a male reproductive system. What creates the specificity? So then you have to go back to the original discussion here. If the DNA binding is the same between these receptors, what's left to dictate specificity? Transcription factor binding of the receptor and steroid binding. And it turns out these two are very powerful in their ability to mediate a response or trigger a response. So you have 4 receptors bind to the same DNA, or very similar DNA, yet you have the biological specificity because of the transcription factor binding and the steroid binding. Does everybody see that? Okay, so then any questions? Okay, so the DNA, yes? Yes, so say you have a gene that's testosterone or androgen regulated. Its DNA binding site will be the same as a glucocorticoid receptor regulated gene. However, the constellation of transcription factors that might bind to it are different, and the steroids that might bind to it are different, so you have specificity. In fact, some people in the field believe the DNA binding simply gets the receptors in the right location, and all the real stuff happens with the protein transcription factor interactions, really mediates the response. That's more philosophy, but it's an interesting point of view. So as you can imagine, say you're looking at various syndromes physiologically in humans. You could predict changes in DNA binding, changes in steroid binding, and changes in transcription factor binding, right? It's not that off to think about that, right? That's exactly right. So it turns out there is a syndrome that in fact has this. All the above, I should say. And it's called androgen insensitivity syndrome, or AIS. In fetal development, if you're a male, you get a shot at testosterone or androgens early on in fetal development. If you properly detect it, you look like a male in terms of your external genitalia. However, if you're defective in your ability to sense androgens early in fetal development, even though you're a genetic male, your phenotype externally, you look like a female. It's called AIS. And there are many examples of AIS out there. These are people that, this is not a fatal disease, or fatal syndrome, I should say. And I saw a seminar about 10, 15 years ago that was speculating that approximately 10% of the female supermodels are actually AIS males. And the reason is the phenotype is great muscle tone, very little body fat, and sort of an androgynous look. Okay? Which, if you think about it, in fact, is how a lot of these supermodels look. I have no idea what the current numbers are. This was a seminar I saw about 10, 15 years ago. So these are individuals that are out there that live And people with AIS, in fact, have a spectrum of phenotypes, all the way from looking like female to almost looking male, or almost male. They're sterile males at the lower, at the less severe parts of the syndrome. Now, what they did is, I tell you from a molecular standpoint, yes, you need the steroid binding region, the transcription factor binding, the DNA binding to be functional in terms of steroid receptors, right? Well, here's a whole bunch of natural, basically, mutations in the population that, in fact, are defective. So the question is, where might they be defective? And guess what? All of the mutations are in the steroid binding region, the transcription factor binding region, and in the DNA binding region of the androgen receptor. Every one of these mutations track to these three areas. Mutations not in those areas have no phenotype change. So this is like almost like a medical correlation, a clinical correlation, that these three regions are important. The fourth kind of mutation here, by the way, is expression. Remember, these receptors are proteins, and they have promoters and genes. And the other kind of mutation is in the expression of the androgen receptor. Okay? So again, it's such an important syndrome that there's an episode of House in the old days of ER. You guys watch ER from a long time ago? One of my favorite all-time shows. There's several episodes based on AIS individuals coming in, and they thought they had a tumor in one side of their body. It's a female, 15 years old. Tumor on one side, they found a tumor on the other side. Turned out to be the testicles that never descended. Okay, so it's actually a real part of the syndrome. Okay, and those of you going into medicine will possibly see this later on in patients and so forth. But what it is, it's the loss of the ability to sense androgen at a certain point in time in field development. Okay, and from our standpoint, it shows how important all three regions are. Yes, question. Yes, because they're male on the inside, even though they're female on the outside. And some of them come up to males, and some of them right in the middle here are almost hermaphrodites. They have both male and female genitalia. Okay, because some forms of the syndrome are not as severe as other. So it turns out that, just as an aside, the transcription factor interactions are multiple. So you can have a mutation there that only has a little bit of an effect. So those individuals are here. But if you have mutations that have a bigger effect, they look more like female in terms of external genitalia. Okay? So you guys, I'm sure you'll study this in medical school. It's always a good one in terms of hormone action. Okay? So these are intracellular hormones, the steroids. There's like another piece of chalk here. And you also have cell surface. We're going to shift gears now to cell surface receptors. And we'll do this over, I guess, here. This is good. Oh, this is better. A lot of your protein hormones, we've already talked about some. CRF, ACTH, we'll talk about LH and GnRH next week, have protein hormones. And some peptides also, epinephrine being an example, are too big. Steroids are small, greasy molecules. They glow right into the cells. The protein hormones can't do this. So these guys interact with cell surface receptors. And you have an extracellular side. This is the outside or extracellular, and the inside of the cell. So the hormone binding is on the outside. It goes to a transmembrane region. On the inside is where all the cell signaling takes place. So some of the changes that you see are receptor-receptor interactions and receptor-protein interactions, such as dimerization, clustering we'll get to. And because of this, kicks on a response. So the hormone binds, you get a change in the receptor, you kick on a response. And what causes the response are a bunch of signaling molecules, or signaling events, I should say, involving changes in protein-protein interactions, involving changes in protein phosphorylation, secondary messengers such as cyclic AMP, changes in ions, and a big one is changes in enzyme activity. So as a result of the hormone binding the receptor, this signal gets transduced through the plasma membrane, and you get all these changes inside the cell that involve a variety of signaling events that we'll touch on. The other issue about this is that these receptors, being on the plasma membrane, have what's called free lateral diffusion. I should say free-ish lateral diffusion. In that here's like a cross-section of the plasma membrane, and these receptors can run up and down. So if you think of an ice cube in a tray of water, that ice cube can go back and forth pretty easily. And if you've got several receptors there, they're moving around. Say the hormone comes in and binds. That dimer creates a very different molecular consequence than as if these receptors weren't dimerizing. So here they're inactive, the hormone binds, they'll become active, or some other molecule comes in and binds. It's because of this free lateral diffusion across the membrane allows this to happen. Okay, questions on that point? Okay. These are the receptors. Yeah. And generally speaking, I'll use erasers as receptors. Okay, unless told otherwise. Okay. So where we're going to go with this? Well, we can't talk about every protein hormone receptor. I talk, again, I have a whole course on this kind of stuff. But we'll talk about just briefly glucose homeostasis. We're going to compare insulin to glucagon. So I think we'll take the last 5 minutes and just give you the overall pathway. So how many of you had breakfast this morning? Oh, good. Some years it's almost nobody. That's good. So when you eat, your glucose levels go up. And what this does, it kicks on in the beta islet cells of the pancreas. These are dispersed out the pancreas. This causes a depolarization of the membranes. It closes a sodium channel, a potassium channel. You depolarize the membranes, and you bring in calcium. And there's high levels of glucose. And what this does is it kicks on the secretion of insulin from the beta islet cell. So this action potential or depolarization brings in calcium, and you get insulin secretion. Now, what does every self-respecting target cell must have? A receptor, exactly right. So the target for insulin is the liver. And there's a receptor for the liver. And when that binds, the liver transports in, or should I say takes in or transports in, takes in and uses or oxidizes glucose. As a result, you reduce the glucose levels in your blood. Okay? So those of you who ate this morning, depending on when you ate, are somewhere in this region here. You've taken in your glucose, you're storing it, and you're feeling pretty good about it. How many of you did not eat breakfast this morning? That's still quite a few. Okay. Well, you guys have a low blood glucose. Most of you. Okay, unless you ate at 5 in the morning or before you went to bed. When I was in college, I had 4 meals. I'd be up at 4 or 5 in the morning. Never made it to an 8 o'clock class that well, but I definitely changed my glucose levels. Shouldn't admit that. Okay, so low blood glucose, and this kicks on. This stimulates the alpha islet cells of the pancreas. Okay, and the mechanism is a little unclear, but probably involved in some sort of action potential change, a threshold of glucose, and it's still controversial how this exactly works. It's much better known for beta islet cells, but probably some sort of action, depolarization of the membranes. This kicks on an enzyme called a hormone called glucagon. Easy to remember, glucagon, right? This is how it was named, I kid you not, because it's low blood glucose, right? The glucose is low. So when the glucose is gone, glucagon gets produced, and what does every self-respecting target tissue must have? A receptor. Well, the receptor for this one is also in the liver. Livers are good things to have. And then the liver then starts synthesizing and secreting glucose, which then increases your glucose levels. And this is glucose homeostasis. I bring this up because what we're going to go into a little more detailed is the mechanism of action of insulin, which represents one important class of protein hormone receptors, and glucagon, which represents another important class of hormone receptors. We're going to look at the molecular mechanism. Okay, any questions? Yes? How does what? Say it louder. Oh, how does high levels of glucose increase calcium? What happens is high levels of glucose depolarizes the membrane. You stop potassium channels. At the same time, you open up a calcium channel. But the bottom line is if calcium triggers release. So if you take calcium, inject it into a beta islet cell, you'll secrete insulin. So calcium is the trigger. It's the way you get there. Okay, have a great weekend, and we'll see you on Monday morning.